is the main factor that determines a species if it is male or female during embryonic development. We have TSD is mostly found throughout reptile clades, and it has different forms. So here on the x-axis, we have temperature, and then on the y, we have proportion male. We have different types. So type A, we have proportion males being found at more cooler temperatures. Type B, we found males at warmer temperatures, and type C, we see males are being produced at intermediate temperatures. So a big puzzle in evolution is the adaptive significance for TSD. How and why did this evolve? So a big idea of how is the Chernoff-Bull model. So this basically states as TSD should evolve if incubation temperature is affecting male and female fitness differently. But what is the mechanism for TSD? Like, why would this evolve? Has been elusive to figure out. The major problems with understanding TSD is laboratory experiments don't really reflect environment very well. Two, a lot of these organisms are very long-lived. For example, this is Jonathan right here. He is an island tortoise. That picture was taken in 1882. That picture in the right corner was taken in 2022. So, like, you need multiple scientists, their whole career is just to follow one organism throughout its entire life. And then three, we even see variation within clades. So painted turtles and common snapping turtles occupy the same range, same environments. They're both turtles, and we see two very different types of temperature-dependent sex termination. So one way of looking at TSD is maybe that temperature is a dominant stressor. And the eggs are incubating or reacting different to stress, with just temperature being this dominant stressor. And there is evidence to suggest that other external factors are also playing into that, that variation of sex ratio. Such as CO2 and desiccation. There's been a few studies that shown that these factors can skew sex ratio. And then we see in the genetics and the hormones of these species, this can also play into variation of sex ratio, and also there is plasticity and individual sensitivity to each one of these variables. So if you take out temperature out of the equation, theoretically, these other factors should be more pronounced, and you should be able to see their effects more clearly. The best place or way to see these effects is at the pivotal temperature of a species during embryonic development. And this is when you, at this specific temperature, you see Fisherian sex ratio, a 50-50 sex ratio for those species. So at incubating turtles at that specific temperature, you should be able to see these different effects of these different variables. So classic Trivers-Willard is originally a hypothesis for mammals, looking at how females will bias offspring to have their offspring be the best competitors as possible to breed. If you think of deer that have this high amount of male-male competition, there's going to be a few males that dominate the population and mates. So if you're a mother in good condition, to increase your own fitness, it's better to have a good conditioned male. If you aren't in very good condition, it makes sense to have a female, because all the females are going to get to mate regardless of their condition. So looking at stress and reptilian TSD, we could replace maternal condition with incubation condition, and where male fitness is being more sensitive to environmental conditions and stress. This is particularly specific to crocodiles and turtles. So a good place to start this, or where I started looking at this, was I wanted to look at the genetics to see if there's any internal factors that might be skewing sex ratio to look at Trivers-Willard. And a good place to start is genetic inbreeding. So inbreeding increases expression of deleterious recessive alleles. It's a known fact if you increase inbreeding, there's also a relationship with decreased condition and fitness, along with insensitivity to environmental stress. Based on Trivers-Willard, competitive males would be likely to be more outbred for competition reasons. So for my research question, does inbreeding affect sex ratio with the species with TSD? My objective was to calculate inbreeding coefficients for a population and incubate these eggs at a pivotal temperature constantly. And then I was predicting that with increased inbreeding within my turtle hatchlings, we would see a more female phenotype. So my research site was in central Ontario at Algonquin Provincial Park. My study organism was the Midland painted turtle. What's cool about this population specifically, in Algonquin, we've been following this turtle population since the 70s, and we have all of this phenotypic data on them. Also, this is the marginal range of the species and the northernmost ranges, so it makes a good model population to look at. So my research design, in June, I would go up to Algonquin Park during the nesting season. And then I collected about 150 turtle nests, and I randomly selected 3 eggs per nest. I then incubated those eggs at the pivotal temperature of 27.3 degrees Celsius. I then sexed each hatchling turtle and then took blood samples from all my hatchlings along with most of all the adults in the wild population. And then I genotyped all individuals using diversity arrays technology in Australia, using SNPs. So for my genetic and statistical analysis, I genotyped the SNP data and analyzed this using the DART-R package in R. I did an immense filtering process where I removed all the monomorphic loci and all loci that were not within Hardy-Weinberg equilibrium. I calculated inbreeding coefficients using observed and expected heterozygosity. And then I looked at the relationship using a generalized linear mix model, looking at a relationship between sex ratio and inbreeding coefficients, along with using maternal ID and container ID as my random effects. So in summary for the results for our population, we were able to collect 568 adults from the wild population, and we incubated about 500 hatchlings. After our filtering process of 87,000 loci, we filtered that down to about 5,500 loci for our genetic diversity analysis. And then our population mean heterozygosity was about 0.23, and then we found a mean inbreeding of close to 0. So very outbred population. And then our sex ratio of our hatchlings was about 58% male, 42% female. So for my main results of this linear mixed model curve, I have I have inbreeding on the x-axis, proportion male on the y. We found that male genetic diversity was slightly more genetically diverse than females, albeit this was not statistically significant. So does genetic variation affect sex ratio? From these results, no, genetic diversity at the pivotal temperature does not affect sex ratio in painted turtles. Again, we did find that males were more genetically diverse, but this was not statistically significant. We can neither support or rule out the Trivers-Willard hypothesis based on these results. And there's some other hypotheses that kind of go along with Trivers-Willard that I can discuss later about that, but for time's sake, I'll just leave it at that. I do want to note something kind of interesting. Compare my study to other studies around the regional area. We found in Michigan that the population was not very genetically diverse at all. In Algonquin, we do see a large amount of genetic diversity. And then as you go towards the coast, we see an enormous amount of genetic diversity. This might be because population's closer to the Great Lakes region. There's a lot more gene flow, so there's less amount of differentiation. But this is something that I might look at in more detail in the future. The plans for this study next, I want to do a GWAS analysis looking to see if there's specific SNPs associated with certain phenotypes, one potentially being sex. And then other ongoing studies I'm doing is I'm doing an experiment looking at how CO2 affects sex ratio, along with how CO2 might also be affecting stress hormones within hatchlings. And then I'm also interested in testing another assumption of Trivers-Willard that only a few males should be theoretically getting to mate in the population. We have all these hatchlings, we have all the blood samples from the wild population. So theoretically, I can figure out if only a few males are mating in that population and what phenotypes are actually making them successful. I'd like to thank my lab, the Rollinson Lab, my advisor now Rollinson, Algonquin Wildlife Research Station, and EUB, University of Toronto. Thank you. Questions? Yes.
Thanks for a great talk. I was wondering if you could tell us more about how you sexed the turtles and whether you saw any potential instances of intersex individuals.
Could you repeat that again?
Yeah, I'm just wondering, sorry, could you tell us more about how you sexed the turtles and whether, I'm just curious if you might have seen any potential intersex individuals?
Yeah, that's a really good question. So the most accurate way to sex turtles, hatchling turtles, is via dissection. So I would actually have to look at their gonads directly. And yes, there are definitely many cases where I would find intersex with one gonad being a classic female-shaped gonad and the other one being classic male. So yes, that did show up.
Approximate proportions? Or if you
like a percentage of the amount? Yeah. I would say maybe like 3 or 4%. So it wasn't out of this norm to see it. And typically I would either remove that individual from the analysis, or I would estimate whether it's likely more male or female based on the gonad shapes. Yeah.
Okay, thank you. You might want to stick those back in a separate category because that is really interesting.
Yeah, that was a thought process. It's just the sample size is a little low. Yeah. Any other questions?
I'm just wondering, have you looked at maybe noise or vibration as a proxy for stress?
I have not, but that's something I could potentially do in the future. Yeah. All right. Thank y'all so much.
So next up, we have another one from the Rawlinson Lab at the University of Toronto. Please welcome Emile Watanabe, who's going to talk to us about sexual differences in acoustic response of snapping turtles to a perceived conspecific.
Hello, thank you for having me. So as Andrea mentioned, my name is Emile, and I'll be talking about acoustic communication in the common snapping turtle. So for most of its history, the study of acoustic communication has primarily focused on birds and mammals. And intuitively, if you think about it, it makes sense. If I asked you, think of an animal that vocalizes, you'd probably think bird. It's common, you hear them all the time, right? And so scientists thought this as well for quite some time. However, in the last 50 years or so, there's been this diversifying interest in looking at other taxa that might vocalize, such as fish or reptiles, in hopes of diversifying our understanding of the evolutionary role of vertebrate communication. And so, naturally, this wouldn't be CHS if we weren't talking about herpetofauna. And so, in fact, there's at least 50 species of turtle that are known to vocalize across life stages, as early as embryos in the egg even, through juvenile stages and then into adulthood. Now, because Chelonian acoustic communication is a relatively nascent field, the function of this communication in many species and even their vocal ranges is still misunderstood. One such turtle that you might know and love already is the common snapping turtle, whose adult vocal range and its function, coincidentally, are still unknown. The snapping turtle is a solitary benthic species and the largest freshwater turtle here in Canada. And so intra-species interactions throughout the year are relatively few. However, during the mating season from May to August, these interactions are increased and generally come in about two forms, either male-male combat or reproduction, right? Union of male and female, and so on. Now, you might wonder, one, does the snapping turtle vocalize like some other turtles? And two, where might it utilize this acoustic communication? Well, based on the literature, there are kind of two large pathways for which we can kind of guess where turtles might use acoustic communication. The first one being sexual selection. Males may be advertising their physical condition, reproductive condition, through their acoustic traits. And so this is actually seen in tortoises, in Hermann's tortoises, where shorter and more frequent calls are correlated with higher red blood cell count, so a physiological marker of condition, and higher mating success. And so it may be that, just like the tortoises, snapping turtles are vocalizing to advertise their condition in a similar manner by some vocal traits or with that varying vocal abundance. Now, alternatively, turtles may be vocalizing to indicate territoriality, which just refers to defending a fixed space for access to mates. However, true territoriality has not been documented in turtles. And while there are agonistic encounters that are common in snapping turtles, they appear to be for access to basking sites or to foraging sites and not actually for access to mates, therefore not meeting that definition of true territoriality. And so that brings me to the goals of our study, which were first to document the full adult vocal range of the snapping turtle, and then discuss where it might use that vocal range. And so we thought to base ourselves off of Hermann's tortoises and explore the effect of sex and then the presence of another turtle, so a social context, on vocal abundance and vocal traits. So similar to those Hermann's tortoises, shorter duration as in a vocal trait, and more frequent calls, abundance, were correlated with that mating success. So this study was conducted in Algonquin Provincial Park in central Ontario in July of 2023. And we caught 17 snapping turtles, 8 males and 8 females, and 1 juvenile, which we did not use for the study. And for each turtle, we put them through this experimental procedure, one by one, always in this same order. Turtles were first placed into a 1,000-liter cattle tank filled with naturally murky lake water and substrate and left undisturbed for 24 hours, all the while being recorded with a hydrophone. The following morning, we moved turtles to an elongated clear water tank filled with UV sterilized groundwater and let them acclimate for 1 hour, again being recorded. We then introduced a 25-pound circular gray bench press weight in the tank to act as a control. Once again, let them sit with it for an hour, being recorded, and then strapped a 3D printed turtle model to that same weight, such that the turtle could see the weight and the model in the same frame, once again, leaving it for an hour being recorded. And so, as you can see, that's our little turtle model in the corner. Oops, not quite. So then we had this acoustic data, and we decided to subsample it, only taking the data from the clear water tank. So that's acclimation, control with the weight, and then the model. And we use kaleidoscope lights analysis software to manually identify vocalizations through this acoustic data, therefore quantifying their abundance, and then using that same software, determining their vocal traits, such as duration, dominant frequency, number of syllables, and so on. And so then finally, we ran generalized linear mixed models to explore the effect of sex and then the treatment on vocal abundance and the vocal traits. So first, what did we find? The snapping turtle in its adult form produces at least 4 distinct types of vocalizations across 2 different clades, hissing and growling. His clades were determined based on these vocal traits, so primarily dominant frequency, duration, and number of syllables. Types 1 and 2 in the top are your hisses, and then types 3 and 4 in the bottom are your growls. Globally, hissing tended to be of a higher dominant frequency, slightly shorter duration, and always of 1 syllable. Syllables being the chunks that you can see in the spectrograms. So in the type 3, you can clearly see these 2 separate chunks. And then growling, slightly lower dominant frequency, slightly longer duration by a few milliseconds, and then either 1 or 2 syllables. Now, for our results on the effect of treatment and sex on vocal abundance. First, we found that when exposed to the model, males are vocalizing significantly more across hissing and growling. And inversely, females are actually vocalizing significantly less when faced in that model as compared to that control disturbance. So, well, what might this mean? For the males, this does support the hypothesis that they may be advertising condition. So, similar to Hermann's tortoises, that increased vocal abundance, where it was correlated with higher mating success, these snapping turtles are increasing their vocal abundance in response to a conspecific. Now, alternatively, they may be vocalizing more just from an agonistic perspective. They may be seeing that model as a threat and just being, I guess, upset at it. Although we didn't actually document any instances of aggressive behavior towards the model, no scratching, no biting, which are characteristic of male-male combat. Now, for the females, why might they be vocalizing less? Well, females are actually known to be wounded in bouts of sexual coercion by males. And so if the females are worried about being injured, then they may be essentially making themselves inconspicuous to the presence of another turtle that they assume is male. Now, this is essentially a mating avoidance behavior. And if so, then it associates or it implies that there is a cost to mating for females, which could be associated with time of year or energetics. In fact, snapping turtle females can store viable sperm for years after a single mating. And so if a female has viable sperm, there's no need to go through the risk of mating and the risk of injury through this coercion. Now, as far as our vocal traits and the effect of sex and treatment, I'm going to direct your attention first to the central panel, which is the effect of sex. We unfortunately don't see any variation in vocal traits across sex. There does appear to be slightly more variation in females with the large black oval in the middle. So there's a little bit more spread there, but it's not statistically significant. Now, on the panel closest to me, the effect of treatment, we do see an effect. The vocal traits are significantly different in both sexes when faced with the model, the plastic turtle, as compared to that control. Now, as I mentioned, vocal traits don't seem to vary with sex, which globally means that both sexes are producing the same types of vocalizations. There are no sex-specific vocalizations. However, both sexes appear to be modifying their vocal traits in response to that turtle. So for males, once again, this does support the hypothesis of advertising condition. Just like Hermann's tortoises, where duration was correlated with higher mating success, In our snapping turtles, the traits that saw the most variation across model and weight treatment were duration and number of harmonics. So these may be the traits by which snapping turtles are advertised in condition. Now, it's really exciting to talk about these potential results and the potential function of acoustic communication, but to draw some conclusions, we'd have to experimentally manipulate these variables, which we did not do in our study. What I can tell you definitively is that acoustic communication is used in between sex interactions in snapping turtles, and that there are indeed sex-specific differences in these behavioral responses to conspecific in a social setting. So that's all I have for you. Thank you very much for your attention. I'd like to thank my supervisors, Claudia Lacroix and Nara Rollinson, for the support in this project, to my field team, without whom none of this would have been possible, and finally to the Algonquin Wildlife Research Station, who housed us, fed us, and created this profoundly positive working environment that is a cherished memory and will continue to be so. Thank you very much. Yes, please.
Thanks, that was really cool. I was just wondering, did you look at this at all at like different times of the year? Like, and did you see any difference in trends like at different parts of the breeding season, for example?
It's a great question. So unfortunately, no. We only conducted these trials in July, so kind of near the end of the mating season, so after nesting, which occurs primarily in June. And so it would really be interesting in future studies to do so. Our concern was if we disturb the population of snapping turtles before the nesting season, that they might just leave our study site. And as Robin mentioned in his presentation, which was the same study site, we've been monitoring these turtles for like 70 years. And so it would be a huge loss for them to leave and move. And so we kind of wanted just to avoid that risk. But if there were a population that could do so and someone could replicate this study, perhaps in May, prior to nesting, maybe even in June during nesting, that would be really interesting. Yeah.
Cool, thank you.
I've
Yeah.
actually got a question online, if I can interrupt. So it's someone saying it's fascinating, Cheryl saying fascinating, certainly leads to a lot of new questions. Is there a next step to this research?
It's a great question. So as I mentioned, I'd really love to do some form of experimental manipulation of these traits, right? Because we can't draw a conclusion from saying, okay, these turtles are vocalizing and it's different with this plastic turtle, but it's still a model, right? So there are no behavioral cues, there are no pheromones, there's nothing there, and the turtle is not really interacting. So can we say that they recognize it as another turtle? Yes, based on their vocal traits and their vocal abundance. But it'd be really interesting to see how it interacts with a real turtle. So that's potentially one route with which we could elaborate. Now, as far as manipulating these traits, we now have this acoustic data. So you could essentially design a playback experiment where we had, these vocal traits and we modified dominant frequency, duration, number of harmonics even, and see if that elicits a different response to those turtles. But yeah, if someone's interested, come talk to me. Yes, please.
So you have a single size of model, I assume?
Yes.
And so you have information on the size of the turtle that was responding to the model?
Yes.
So I don't know if I missed it, but do you have any insight on whether there's greater number of vocalizations or fewer, depending on the size asymmetry between your model and the actual test subject?
It's a great question. Initially, when we designed the study, we had body size as a proxy for conditions. And so that was the initial hypothesis, but we ended up removing that from our statistical analyses. Off the top of my head, I actually don't know whether body size within a sex affected number of vocalizations. I can tell you that males vocalized more globally, but males also just tend to be larger. Yeah, no, I'd have to look at that. Thank you.
We still have time for like one more question if anybody wants to.
Yes,
take it.
please.
Perfect.
What was the original thought behind the 24-hour murky water tank? Is there like a reason why you thought to do that? Obviously, you didn't have time to analyze it, but is there a thought?
Yeah, absolutely. So it was an ambitious project, and we had lots of ideas. And honestly, I think I was overwhelmed by the sheer amount of acoustic data that we got. And so we picked the three trials having that acclimation in the similar conditions with that control. But initially, the 24 hours was to see, one, we're replicating a natural environment, right? So there's the like water that we pulled from the lake, sand that we pulled from the lake, and we filled this tank with it, as opposed to just water, to water from a hose. So, I mean, there's lots of questions that could come out of that. One being time of day, for example. We've now got 17 turtles in a tank, natural environment, so minerality of the water, maybe potentially other chemical cues from species already present in that water. You could look at number of vocalizations per day, or maybe sexes are differently active across different times of day. You could even compare the water, the lake water, to the clear water and the acclimation of both, for example, to see, does the tank condition affect vocalization? Are they potentially just stressed in an environment they don't recognize? It's actually the product of a new project from our lab. Another undergrad is taking it on, and so we'll hopefully maybe next CHS there's a talk on it, but we'll see. Yeah, absolutely. Okay, thank you.
So this is our last speaker for this session for today, Charlotte Turenne. So I went very French with it, but that's okay. From Carleton University, who's going to talk to us about personality in spiny softshell turtles. So welcome, Charlotte.
Hey, hi everyone. Tiny turtles with big personalities. My name's Charlotte Turenne. I'm a student from Carleton University, here today to present to you my undergrad thesis that I did under the supervision of Dr. Christina Davey and Dr. Sean Boyle. And for this project, I was investigating the presence of personality traits in neonatal spiny softshells, and their Latin name is Apalone spinifera. Okay, so I just wanted to start by just talking a little bit about animal personality as a concept. So this describes the individual variations in behaviors that remain consistent over time and across different environments, so just different contexts. And within this field of study, we have 5 general personality traits that we assign to animals. So we have shyness, boldness, exploration, avoidance, activity, aggressiveness, and sociality. You may notice that some of these are hyphenated, so shyness, boldness, and exploration avoidance. This is just because behaviors can exist on a continuum. So the same behavior is being expressed just in one direction or the other direction. So sociality is the personality trait that I'm actually focusing on in my project, and it can be most simply defined as an animal's tendency to associate with, live with, or just gravitate towards other conspecifics. Now, reptiles were not really historically viewed as social animals, but there's been a lot of interesting research published lately that would suggest otherwise. I'm sure most of you guys would disagree with that as well. And we see examples of sociality in reptiles in nature all the time. So whether that's painted turtles basking on a log together for thermoregulation, male garter snakes in a mating aggregation, the 5-line skink displaying parental care, or the snapping turtles in a feeding aggregation. Now, if we take a look at the evolutionary history of turtles, we can see that they're most closely related to a group called archosaurs, which consists of modern birds and crocodiles. This relationship is based off of molecular and genetic evidence suggesting that they have a common ancestor. And birds and crocodiles, as we know them, are pretty social beings, and they display high degrees of parental care, whereas turtles do not. So this provides an interesting study species for a study on reptile sociality. So in order to contribute to the growing literature on reptile personality and sociality, I was interested in investigating if freshwater turtles showed any evidence of measurable personality traits upon hatching, with a specific emphasis on their social behavior. So to do this, I tested the hypothesis that hatchling spiny softshell turtles would display variable personalities through the prediction that they would exhibit repeatable social behaviors. Now, I really want to emphasize here that repeatability is absolutely crucial for this concept. Behaviors related to personality traits must be repeatable within an individual in order for that personality trait to be considered present. So to do this, I conducted controlled behavioral trials to test repeatability of social behaviors. So first off, we collected nests and we incubated them as part of a larger Head Start program to prevent nest depredation of the already very endangered spiny softshell turtle in southwestern Ontario. We collected them and incubated them. And then once hatchlings emerged, we transferred them into a little arena, as I call it, which is just a little plastic container, as you can see up on the screen. We marked hatchlings with Sharpie marker number on their carapace for identification throughout the video trials. And they underwent 3 successive trials under identical conditions, lasting 5 minutes in duration. We had a total of 13 groups with 3 to 5 clutch mates per group for a total of 56 individuals used in the study. Now, in order to score their behaviors, I developed an ethogram. So the ethogram consists of a list of behaviors and their corresponding definitions. Now, for simplicity's sake, I didn't include the long wordy definitions, just some cute infographics to give you an idea of what these behaviors may look like. So my final ethogram contained 3 behavioral categories. These are my contact behaviors, my response to contact behaviors, and my locomotion behaviors. So the behaviors in the contact and response to contact categories are all attributed to sociality, and repeatability of these behaviors would be an indication of social personality. And the same goes for the locomotion category. However, that one is attributed to activity, another personality trait. So I've included some short clips here to give you an idea of what I was really looking for in the video trials, also because baby soft shells are adorable and super funny. So the first one is a short face-to-face interaction leading to a period of stationary contact between hatchlings number 1 and 3. I'm not sure how well you can see the numbers marked on them, but you'll see once they start moving. And then they're just hanging out for a little bit. It's pretty cute. Next up, hatchling number 2 climbs on top of hatchling number 3, to which hatchling number 3 displays no reaction to this form of physical contact. Yeah, and they're just happy there for a little bit. So this is just to give you an idea of really how simple the behaviors are. So to analyze the data, I went ahead and I used linear mixed effects models using the RPT-R package in R, and I calculated the interclass correlation coefficient, which is represented by an R value. So R values represent quantified levels of repeatability. And in my model, the R value uses variance to quantify what proportion of repeatability can be attributed to differences between individual hatchlings. So R values are a scale, and as you can see, in general, higher R values indicate better or higher levels of repeatability, and they range from 0 to 1. So I went ahead and I ran models for all 12 of the behaviors in order to test repeatability. And I wanted to mention here that we also ran this model using sex as our predictor variable, and we found negligible levels of repeatability, with a third of them being 0. So the following results will show the levels of repeatability that is attributed to individual differences. Okay, so over on the x-axis, we have our R values ranging from 0 to 1, with a guideline for how to interpret them down below. And then our y-axis, I have all 12 of my behaviors. So first up are the locomotion behaviors. These ones showed highest repeatability overall, all above 0.6. So these ones are showing good repeatability. These ones, again, are not an indication of sociality, but they are likely an indication of activity, another one of our 5 personality traits. Next up, response to contact. These ones showed pretty low repeatability overall, all falling within the poor to fair levels of repeatability. And then our contact behaviors, these ones range from poor to moderate. There was a pretty big jump between them, but we have incidental contact, stationary contact, and climb partner, all falling between 0.4 and 0.6. So overall, half of our behaviors fall within the moderate to good levels of repeatability. So the good to moderate levels of repeatability that we found in the behaviors associated with sociality and activity indicate that hatchling spiny softshells do indeed display variable personality traits to some extent. So since the individuals in our study were incubated and they hatched in captivity, we conclude that any personality traits that were detected can be attributed to genetic factors, or rather innate characteristics. So since these individuals haven't really spent any time in the wild, they wouldn't have had time to adapt their behavior to any changing environmental conditions. And this is important because behavior has implications for fitness, and it helps us better understand a species overall. So my study species is endangered in Canada, as we all know, so more behavioral studies would definitely be useful for getting kind of the whole picture. That being said, I do think that future research should focus on understanding what factors might influence changes in turtle personality traits over time. So these factors could be things like age, health, environmental stressors and environmental change, and social interactions. And finally, I think it would be interesting to know how differences in personality contribute to survival and overall fitness, both for individuals and for an entire species. And then I'd just like to finish off with some acknowledgements. So I wanted to acknowledge that our fieldwork was conducted in the traditional and treaty territories of the Adirondack and Mississauga nations. I want to extend a huge thank you to Christina Davey and Sean Boyle for their ongoing support with this project and for their patience with me in my first sort of research experience. And then the 2023 Wetlands and Reptiles field team, they helped a lot with egg collection, monitoring the eggs throughout incubation, as well as the behavioral trials. So thank you so much for your attention, and I'm happy to take any questions.
Yeah, great talk. This is super cool. I'm really curious about the last question that you're talking about, like future questions or future directions. What do you think the survival or fitness benefits would be of having these different personalities?
Yeah, I think this kind of relates to like the workshop we did yesterday on genetics. So within a population, it's really important for individuals to have variable personality traits so that they can persist throughout environmental change. And another benefit to that is if we do understand this and we can see in like vulnerable populations, let's say there's environmental change and we see that these individuals aren't generally social animals and we start to notice them in these feeding aggregations, we can think about whether or not there's changes to food availability and stuff like that. At the back.
Hi, great presentation. Really interesting. So my question is, do you think that the results would have varied if they were placed into a controlled environment that wasn't plastic? Like if you replicated sand or water or murky water or anything like that, like similar to their habitat, and whether or not that would have changed the results?
I would love to know. That's definitely a possibility, especially because like with spiny softshells, they do tend to bury themselves under the sand and kind of hang out. That's like kind of how we see them in the wild. So I think it would be interesting to see if they did that. But it was also cool. Like I watched a lot of video trials and throughout there were some individuals that would hide in a corner and really avoid any sort of physical contact. And others would, it seemed to me like they would seek out contact with individuals. So I would love to see if results were different in a different environment. But I do suspect that the individuals that were like making it their goal to like hang out with the other turtles would potentially still be doing that even in their natural habitat. Sure. Anna?
Thanks. This is super cool. I was just curious, and sorry if I missed this in your design, but if the hatchlings in the same experiment were from the same nest, or if you mixed them up?
Yes, exactly. They were from the same nest.
Okay.
And that's interesting too. I would also like to see if results were different if they weren't from the same nest, just because, as we heard before, like there's vocalizations, like sometimes turtles will hatch at the same time and kind of head out all together. So that would be interesting to see if there was maybe less sociality, if they were with individuals that they weren't incubated with, like not their clutch mates. But I think just for simplicity's sake, we kept clutch mates together because we did have to release the eggs where we found them. So in order to not mix up turtles and get them and like release them in the wrong areas, I think that was kind of the reason we did it that way, but it would definitely be interesting to see. Yeah.
Yeah, cool. Thank you.
Yeah, great presentation.
Thank you.
I was wondering, do you know with the contact behavior, is there like some sort of like stress relief effect on the turtle that you know of or read about?
Not that I know of, honestly.
Okay.
I'm not sure. And I didn't see anything really about that in other studies. And there's not too much when it comes to turtle sociality specifically, especially at the neonatal stage. So I honestly don't know.
Okay, fair enough.
Okay, thank you.